Hi, Assalamualaikum. My name is Fatin Najiha from Johor Matriculation College. So today I will present about method in gene cloning. So there is 5 steps in gene cloning that we need to know. So number 1 is isolate the DNA. There are 2 DNA that we are going to isolate. 2 DNA. Number 1 is DNA plasmid from bacterial cell. Typically we use as E. coli. So this is a plasmid. Okay, so number 2 is a DNA that contains gene of interest in a living thing. Okay, we can use this such as animal or plant. Okay, so this is the first step, which is we need to isolate the DNA. So second step is cleave or cut. So this process is we do it in a test tube. Okay, we do it in a test tube. So this is isolated plasmid, and this one is isolated DNA. Okay, we go one by one. First, we go through the isolated plasmid first. Okay, this one is the first one. Okay, so this is what it looks like and inside. In this step, we need to cut. Okay, these 2 enzymes. Both of them will be cut with the same and compatible restriction enzyme. Okay, so this process will produce the complementary sticky end. Okay, I will show you for the first one, which is the plasmid. So it will become like this. It will become like this. Okay, this is where it will be cutted. Here. So here is an open plasmid. We call it as an open plasmid. Okay, so this plasmid is cut using the same restriction enzyme. For example, if you use restriction enzyme EcoR1 to cut open the plasmid, we will also use EcoR1 to cut the target gene into multiple fragments. So we go through the number 2, which is DNA. So number 2, green one. Number 2, okay, this will produce like this, and it will become like this. Okay, this is a target gene. Here. Okay, this is what we call as DNA fragment. So as a result, we will get this target gene, or called as a DNA fragment. Also, this one, the open plasmid one. Okay. So as the result, we will get this and also this. We will get 1 molecule of plasmid, this one, and many molecules of DNA fragment. Gene of interest and the plasmid are now having complementary end, which is sticky end or blunt end. Okay, by using the same restriction enzyme, but in this diagram, the complementary end is a sticky end. Here is a sticky end. Here and here, also here and here is a sticky. So the third step is insertion. Okay, what will happen here is the DNA fragment is inserted into a plasmid. So from this plus with this one. This is what we call as insertion. Okay, this is what we call as insertion. So we insert the target gene into the plasmid. Both the target gene and the plasmid are joined by using DNA ligase. So what is DNA ligase? So DNA ligase is like a glue that bind together two fragments together by forming a phosphodiester bond. Okay. So it will, as the result, you will get a recombinant plasmid and non-recombinant plasmid. Like this. So this one. Like this. This. This actually will produce three. Okay, this one is a recombinant. Okay, this one is a recombinant plasmid. This one is non-recombinant plasmid. Meanwhile, this one is just like plasmid together because it has a sticky end right here and right here. So they just join together without any DNA fragments or target gene that we want. Number 4 is transformation. This is done by returning the plasma into the host cell, and the common host cell of bacteria used is E. coli. So this is recombinant DNA that we have done earlier. We'll get into a host. This is a host cell, which is E. coli. This is a recombinant DNA. Okay, we return the plasmid into the host cell. Okay, so this is a host cell. This is a host cell. Okay, we put it the recombinant DNA inside this. Okay, we put it inside this and treat the bacteria with calcium ions, which is calcium chloride, and brief heat shock to make holes appear in the cell surface membrane and to make them permeable to DNA, allowing the plasmid to enter. Okay, so as a result, there we have 4 types inside the host cell. So as a result, there are three possibilities of the whole cell. So the first one, okay, let me show. This is the first one. Is a whole cell that took up the recombinant DNA. So this is recombinant DNA. Took up the recombinant DNA. It could be with or without the gene of interest. Okay, the second one is plasmid, but with the second one is non-recombinant. So this is the second one, non-recombinant. Because there is no target gene that we want here. And the last one is the whole cell but without plasmid, no plasmid. Okay, next is still under the step 4, but this one is amplification. So amplification, this is a culture, the bacteria. Okay, now all the bacteria will be cultured on an agar medium. The agar medium is supplemented with ampicillin, which is an antibiotic, and also a agar, which is a molecule that resembling lactose. Okay, this technique is called This one is called amplification. Copies to make multiplied copies of target gene. Okay, and now the agar plate will be incubated until the colonies grow. Okay, only cell that took up plasmid. Okay, this is we have 3 results. Okay, but only cell that took up plasmid, which has ampicillin gene, will reproduce and form a colony. Okay, so it will incubated until the colonies grow. Okay, so here we will go through to the last step. Step is screening. It's a blue-white screening. Okay. The function of the blue-white screening is to identify the recombinant DNA. So let's go and look through the colonies inside the bag. So here it's white and blue. Okay, first we're gonna look about blue colonies. Okay, the blue colonies is hook up non-recombinant DNA, non-recombinant plasmid, yeah, same thing, and contain intact lysine and code beta-galactosidase and can hydrolyze X-gal. But for the white one, so this is number 2, which is white colonies, is a recombinant plasmid, recombinant DNA, and like Z is disrupted, cannot hydrolyze X-gal. So at the end of this experiment, we choose the white one, okay, because it have a recombinant DNA in the white one. So we choose it from here, white one, like this.